All right, let's go through some of these single-cell RNA sequencing techniques and what we're trying to do. And so this is a list of things you can do with it, and yeah, it's kind of interesting. So you can understand tissues where there are different types of cells a little bit better if you can see what each cell is making. Because, of course, the DNA doesn't distinguish any cells in your body except for cells of the immune system, because they delete some parts of the DNA. But the RNA is completely unique to each cell type, and even within each cell type, there's a lot of variability. So you can find rare cell types, new cell types, cells that are doing unexpected things, versions of a cell that we know about, but it's making something extra that gives it an extra function, for example. Yeah, you can look how that changes over time, changes when you have disease. Yeah, there's a lot you can do with it, really. So here's that same paragraph, but in pictures. So you can take different populations of cells. You can look within a particular, what we think of as a type of cell, and see, okay, what variability is there actually inside of this group? And you find that, you might think there are just hepatocytes, but there are actually probably several different general versions of any cell type that are making more or less of certain proteins, and each of these probably is playing some role in physiology, which may not have been known without this technique. So you can look for differences between different cell types. So what makes a particular cell type that kind of cell? Which genes do you have to express? Because theoretically, you can express any gene from any cell, but when they're expressed in particular patterns, then they have a phenotype. They look like a neuron. They start to do, make neurotransmitters and big long axons and things like that. But it's all controlled at the level of which genes are being expressed. And then you can also look in a disease state between different tissues. You see a lot of these looking at people who have COVID versus people who don't have COVID. So, what extra cell populations get turned on? What's the immune response look like? What are those immune cells primed to do? Because you can show immune cells are there, but an immune cell being there doesn't necessarily mean anything. It really matters what is the state of expression of lots of different parts that'll tell you if that cell is just a bystander that's going to, wash past and keep going, or if it's something that's going to have a profound effect on the tissue where the infection is. Yeah. And so, single cell expressions. Pretty cool. Yeah. All right, so let's go. So over time, of course, we've got better. The early papers and the early methods were able to do, just a single cell or small groups of cells, like up to 100 cells, and profile what is going on inside of each of those things. And then over time, we developed all these different techniques. You can see these are mostly the names of companies. You can look these things up. The ones that were popular in 2017 have given rise to new ones that come after, but there isn't a cool graphic showing them, so this is the graphic we're going with. The way this works is, the first ones were done absolutely manually by picking up a single cell and pulling it out and breaking it apart and then sequencing all the RNA inside. Then they figured out how to multiplex, so how to essentially pick up several cells and look at all the RNA inside at the same time. Then they figured out these sort of microfluidics, which allow us to do things like Illumina sequencing, which really speeds up the sequencing process. And liquid handling robots that can allow us to sort of automate some parts of this and catch even more data. The current techniques, I think most of the ones over here, are actually versions of this, which is called a Drop-seq, where you have a little droplet of water and enzymes and stuff like that, and you have your cell inside of it. And you'll have other stuff inside of it depending on what you want to do, but you're going to keep each cell separate from the rest in an emulsion. So it'll be a water droplet inside of a bunch of oil, basically. And then they figured out, well, we could take these little water droplets and we could sort them into tiny honeycomb-like wells. so that one droplet fits in each well, and that's great. And then it's sort of like pyrosequencing. You've got your little chamber, and you're going to sequence everything in that chamber. And then since then, they've been doing all kinds of crazy stuff. So the idea of in situ barcoding is, I think, you can actually read out inside of an intact cell what it's making. So those are pretty neat. All right, so what is the procedure? Well, here's a nice picture. And if we move this over just a smidge, there we go, crunch myself into the corner. Here is a generalized way that Drop-seq works. So the companies will have different versions of some of these components, but this is a good enough general schematic that we can just assume that this applies, and then as long as you learn this, then learning other variants won't be as difficult down the road if this ends up being something that you do. So you have these little beads, and the little beads are going to contain lots of oligonucleotide primers, so little chains of DNA. And the chain of DNA is going to have several different regions on it. So on one end, it's going to have a poly-T region, because poly-T is going to bind to poly-A, the Watson-Crick base pair. And since poly-T is going to bind at the end, the poly-A tail of an RNA, it can then be extended this way to copy the entire RNA. So a poly-T covered bead will be effectively a universal primer for any messenger RNA, because all messenger RNAs should have a poly-A tail. Yeah, at least any conventional messenger RNA. Then they're going to have a barcode, and different companies will use different barcodes. The most common one is an 8 nucleotide barcode, which gives you up to 65,000 different options of barcodes. So like every combination of A, C, G, T, and there's 8 of these in a row. And so that's just going to be a unique identifier. So this is going to tell us that this particular, every piece of RNA on this bead is going to have the same barcode. And so we're going to know that they all come from the same cell and the same bead, because you're only going to put one cell onto a bead and sequence everything there. And then the R1 region, this is going to be, it's also called the PCR handle. It's basically a primer that they know the sequence of that will bind to an Illumina plate, so you can do Illumina sequencing. And if you're a little fuzzy, you're saying, what the heck was Illumina sequencing? Go back. There's like a 5-minute video or something like that on it earlier in the course, toward the beginning. I think the maybe second or third section that we went through. But yeah, right now all the readouts on these are with Illumina sequencing. That is the next-gen sequencing platform that is winning or has won. I guess there are some others still out there, but it's definitely winning at the moment. Okay, so you make these beads. Every bead is covered in primers. All the primers have the black part and the blue part the same, and the red part is, I don't remember what the UMI is, but yeah, something else that they can use. There are different versions of this that don't have that extra part right there, which is why that one's a little strange. This is probably specific to the 10x genomics setup. But they all have a barcode in the middle. They all have a reverse transcription, not a reverse transcription, but a PCR handle at the end, like a thing that you can make a primer against. And they all have a poly-T at the other end. Yeah. So there you go. And so different barcode for every single bead. And so if you're thinking, how do we assemble this the first time? Very expensive the first time. But once you've made a batch of each of the 65,000 beads, then putting them together in a little packet of one of each bead, not that bad. You can get a machine to do that. So for the rest of this, you need a special device that uses microfluidics, like little tiny channels where water will flow. And it's going to look at cells. It's going to examine the cells and see what kind of cells they are. And you can do this in a number of ways, but the commonest way is going to be with fluorescently tagged antibodies. Now, we want the cells to be alive and intact, so we don't want the RNA to be broken down or to have problems amplifying it. So yeah, this is going to be with live single cells, and we're going to bind these fluorescently labeled antibodies to surface proteins on these cells. Yeah. So the labeled cells can then be sorted, and so there'll be a laser right over here that will check what's the fluorescence of each cell, or we'll already sort the cells by fluorescence. Then we'll say, okay, from this batch of cells that had these particular 3 proteins on the surface, let's just send them all in here and sequence many members of this cell type. So then we come in here and we're going to merge the cells with the beads so that we end up with only 1 cell per bead, and some beads won't get a cell, so those will just be wasted basically. So we'll collect them, and so there they are, little sort of pods of water with 1 cell and 1 bead inside them. Then the next thing is to burst the cells open, and they can do this with heat or with chemicals. And then they will copy all of the RNA into DNA using these primers, and using RT there is a reverse transcriptase, which is an enzyme that copies RNA and makes a DNA copy of it. And then we'll take the DNA and make a second DNA copy of it. It's very nice. It's like a polymerase that can also do something else. And so each of these little droplets will now have uniquely barcoded, because everything in the droplet will have the same barcode, but the barcodes will be different in each droplet. It'll have differently barcoded DNA. From this point, you can then sort out this DNA. You can say, I only want things of a certain size. I only want a certain type of gene. And so we're not going to learn that part of the protocol, but you can have some sort of sorting based on size or sequence at this stage. And then after that, you're going to basically centrifuge them to get all the water to come together and all the oil to come together. And then it's beautiful. You have these little DNA fragments. They're all loose in there together, but they each have the barcode that they got from the bead they used to be attached to. And because of that, you can then go back in later and sort them out and say, oh yeah, you are all from bead number 1, and so you must have been from cell number 1. And these others are from bead number 2 because they have barcode number 2 on them, so they must have been from cell number 2, just like that. All right. So that's where we'll pause this video. And this method is called Drop-seq, because we're putting our little cell into a little droplet and then bursting it open and amplifying it, and we're going to sequence everything there. The primer we use is going to be poly-T, so that we can pull out all the poly-A sequences from messenger RNAs. We're going to use reverse transcriptase, this is the RT right here, in order to copy the messenger RNAs into cDNA that's attached to the bead. And then you can kind of dissolve the bead or release the DNA from the bead, and each company has its own way of doing that. All right, there you go. And in the next section, we'll do this technique, but with an extra modification that will allow you to quantify the amount of protein as well as the amount of RNA in every cell. So stay tuned.